This is the first video of the urinary system. At the time we approach a system, the first thing that we have to clarify and understand is the function of this system. In this case, the urinary system function can be summarized in only one word, that is regulation. Yes, regulation of the blood volume in plasma, regulation of the waste in plasma that need to be eliminated, regulation of the electrolytes in plasma that need to be in balance, regulation of the blood pH. All these factors are regulated by the urinary system. Before to jump in physiology by itself of the urinary system, let's take a brief review about the anatomy of this system. Yes, the urinary system is made by two kidneys that the function of them is to produce urine. That urine is evacuated into the ureters, and the two ureters send the urine into the urinary bladder. Now, that urinary bladder used to be just a store for urine, and when it's the proper time, that urine can be evacuated using the urethra. Now, the urine is transported in that tubes using peristalsis movement. But also the kidneys are located retroperitoneal, means that they are outside of the peritoneum, and they are located in the back wall of the abdominal cavity and below the diaphragm. Continue with the anatomical structure of the kidneys. Please be knowledgeable about the areas that can be differentiated inside the kidneys. One is the renal cortex, the other one is the renal medulla, and the renal columns. Also, be familiar with the vessels that can get into the kidney. One is the renal artery, and the other one is to collect the waste and take the deoxygenated blood outside of the kidney. And this one is the renal vein. The renal artery is a branch of the descending aorta, and the renal vein is going to drain into the ascending vena cava, that finally ending up in the right side of the heart. As many other internal organs, the urinary system organs are regulated by autonomic nervous system. And in this case, the parasympathetic nervous system is the one that stimulates the urinary system, and the sympathetic nervous system is the one that inhibits the function of the urinary system. Also, the parasympathetic nerves are emerging from the sacrococcygeal area in the spinal cord that emerge into two neurons. The preganglionic, that is very long, and the postganglionic, that is very short. One way that the urinary system has to control the blood volume, and by controlling the blood volume, control the blood pressure, is through the amount of urine that we produce in urination. Now, let's take a look about how we control that urination. Urination and micturition are synonyms, and there are different players in that game. One of the players is the detrusor muscle. The detrusor muscle is the smooth muscle that made the wall of the urinary bladder. The other player is the parasympathetic nervous system, using the parasympathetic neurons, parasympathetic motor neurons. These parasympathetic motor neurons use, like the postganglionic parasympathetic neurons, are using acetylcholine. In this case, the acetylcholine binds with the specific receptor, and this specific receptor is not nicotinic, is muscarinic. And in this specific system, the interaction between acetylcholine with muscarinic receptor is excitatory. In other words, produce a depolarization of the detrusor muscle cells. By depolarization, produce a contraction, and by contraction, we can expel the urine into the urethra. The other player in the control of micturition are two sphincters. In the urethra, either males or females, we have two sphincters. One is internal sphincter that is made by a smooth muscle by which is involuntary. So the internal urethra sphincter is made by a smooth muscle and is involuntary. This one gives you an idea that if it is a smooth muscle and it is involuntary, it's controlled by the autonomic nervous system, either parasympathetic or sympathetic, never simultaneously, one at a time. The other final player is the external urethral sphincter that is made by a skeletal muscle. That skeletal muscle, obviously, is voluntary, is the one that we control, is the one that we keep contracted in order to avoid the urination, and is controlled by somatic motor neuron. In this graphic, you can visualize that the postganglionic parasympathetic neuron is the one that makes the synapse with the detrusor muscle cell. In this synapse, this postganglionic parasympathetic neuron use acetylcholine as neurotransmitter. And this acetylcholine travels in the cleft to find the proper receptor. The receptor is muscarinic. Muscarinic receptor is just a receptor. It's not receptor and a channel, just a receptor. But when acetylcholine binds with muscarinic receptor, trigger the opening of the channel nearby. And in this case, opening this channel allows calcium to get into the detrusor muscle cell. Calcium is a positive ion, and like any other positive ion getting into the muscle cell, produce a depolarization. That depolarization is the one that produces a contraction. Now, let's see how those players are synchronize the action in order to control the micturition. In our urinary bladder, we have stretch receptors. These stretch receptors are the ones that sense when that urinary bladder starts filling up with urine, little by little, during the day or during a period after you urinate. Now, that stretch receptors in the urinary bladder send a message to the central nervous system in the sacrum region of the spinal cord. What is informing the central nervous system that the urinary bladder is getting ready to be empty again. Now, we have two reflexes that are working in order to control the micturition. And these two reflexes are the guardian reflex and the voiding reflex. The guardian reflex is like the one that protects us to have an involuntary emptying of the bladder. And in this case is the one that is regulated by the somatic motor neuron that innervate the external urethral sphincter. Meanwhile, we have that neuron, the somatic motor neuron, stimulated. Meanwhile, we have this neuron stimulated. We have the external urethral sphincter contracted and avoiding involuntary emptying of the bladder. This one is the guardian reflex. Now, when we go to the bathroom, because either we have time, we don't want to wait more time, or we are in a rush, at the time we get into the toilet, we have that guardian reflex that is inhibited. In other words, we release the action of the somatic motor neuron, inhibit the action of the motor neuron, and we allow the parasympathetic to take place. And this one is the voiding reflex. The voiding reflex is not just in the internal urethra, it's also in the whole detrusor, because the parasympathetic nervous system produces contraction of the urinary bladder, by which the urine in the bladder is sent into the urethra, and because we have the guardian reflex totally relaxed, we can empty the bladder. Let's be focused in the microscopy structure of the kidneys. And in that microscopy structure, we have to be focused in the nephron, because the nephron is the functional unit of the kidney. That functional unit of the kidney is the one that produces urine. We have 1 million of these nephrons in each kidney, and certainly when they are damaged, produce severe kidney disorders. Now, these nephrons are composed by different structures that, in combination of all together, produce the urine that is evacuated, is dumped into the collecting tube, and from there is emptied into the minor calyx, major calyx, renal pelvis, ureters, urinary bladder, urethra, and outside the body. The blood flow inside the kidneys is also a very important topic. Probably you listen from different classes about the afferent and efferent arteriole of the glomerulus, also glomerulus itself. Well, this afferent arteriole is the finishing of one circuit that is originated in the aorta artery. This aorta artery is getting ascending, is getting the aorta arch, is getting descending, cross the diaphragm, and when it's in the abdominal cavity, that aorta, descending aorta, produce one branch that is the renal artery. That renal artery is the one that is getting into the kidneys and carries oxygenated blood, fresh blood with all the nutrients to irrigate all the cells inside the kidney. Immediately gets into is branching out, getting smaller, and it's always using the renal columns to go through. That branches of the renal artery, they never cross in between the medulla. They are always located in the renal columns. And in the branching out, the finishing of that renal artery is the afferent arteriole. This afferent arteriole is the one that originates the capillary bed. And this capillary bed is a special capillary bed. It's called glomerulus capillaris. When after the capillary bed, that capillary bed produces the efferent arteriole, and this efferent arteriole is the one that originates the other system called peritubular capillaries and vasa recta capillaries. Those capillaries are normal capillaries that are around the different structures that compose the nephron. But certainly from there on are branching in, getting bigger, using the same pathway was used for the arteries, in other words, the renal columns, to merge into the renal vein. And from the renal vein are draining deoxygenated blood into the ascending aorta. The ascending aorta is sending the blood to the right side of the heart. You also have to be very familiar, knowledgeable about the structures that compose the nephron, starting from where they are originated, that is the renal corpuscle. This renal corpuscle is composed by the renal capsule that has a C shape on the top of the graphic, the renal capsule, and the glomerulus capillaris. These two structures compose the renal capsule. From the renal capsule originate a combination of tubes. The first one is the proximal convoluted tube, following by the loop of Henle, and the loop of Henle originate the distal convoluted tube. The distal convoluted tube is draining the fluid into the collecting tube. The collecting tube is draining the product into the minor calyx. Now, keep in consideration where the renal corpuscle and the convoluted tubes are located, always located in the cortex of the nephron. On the contrary, the loop of Henle and the most important part of the collecting tube are located in the renal medulla. Another aspect that you have to consider and also be knowledgeable is about that intricate and dense network of capillaries that are around the nephrons. Starting from the afferent arteriole. As I mentioned before, the afferent arteriole is the ending of the renal artery. The ending of that renal artery is this afferent arteriole that is producing that special capillary bed called glomerulus capillaris. And at the end of the capillary bed, or the glomerulus capillaris, is emerging the efferent arteriole. Now, keep in mind that these glomerulus capillaris are special capillaries. You notice that they don't change the color from blue to red or from red to blue. They remain all the time in red because the blood that is running inside of this capillary bed, the glomerulus, is oxygenated blood. Now, the efferent arteriole, as I mentioned before, originate a red of capillaries, standard capillaries, that are called peritubular capillaries around the distal and proximal convoluted tube, and around the loop of Henle is called vasa recta. Keep in mind the two areas, cortex and medulla, because that one is important information that you need to keep in consideration when we go to the second video. I took the time to summarize in this slide the zones of the nephron and the function of them. As I mentioned before, the nephron is originated in the renal corpuscle, and the renal corpuscle is made by the glomerulus capillaris and the renal capsule. The function of the renal corpuscle is unregulated filtration. When I mention regulated or unregulated, means that in case of unregulated, there are not hormones that influence that filtration. The only reason or the only factor that influence the filtration are particular forces. One is the most important is the hydrostatic pressure or the blood pressure, and the other force is the oncotic pressure or osmolarity pressure or protein pressure. The other zone in the nephron is the proximal convoluted tube, and the function of this proximal convoluted tube is unregulated reabsorption, means that there are no hormones that influence the reabsorption at the proximal convoluted tube. The next zone in the nephron is the renal loop, and in this case the molecules move by active or passive transportation. The renal loop is composed by two areas. One is the descending limb that is coming from the proximal convoluted tube going into the deepness of the medulla. And the function of this descending limb is reabsorption of water by osmosis. Following the descending limb, we have the ascending limb, and this ascending limb goes from the deep of the medulla all the way back to the cortex, and the function is reabsorption of sodium using the sodium-potassium pumps. Notice that I wrote sodium and chloride because in chemistry, sodium is always matching with chloride because uh, where sodium moves, chloride will follow. They have opposite charge, but also sodium moves by sodium-potassium pumps, but chloride move the movement of sodium by passive transportation. The distal convoluted tube is the next zone in the nephron, and in this area, in the distal convoluted tube, the function is regulated secretion and reabsorption by hormones. So in this distal convoluted tube, we have two functions, either secretion or reabsorption. But this action, this movement, is regulated by hormone, specific hormones that I will describe later on. The final zone of the nephron is the collecting tube, and the collecting tube is not used by only one nephron. It's used by really several nephrons close by. But the function of this collecting tube is reabsorption of water by one hormone. In other words, this reabsorption is regulated by a hormone, that is the antidiuretic hormone, in case our physiology needs the use of this hormone. In other words, when you have filtration plus secretion minus reabsorption, we have the total urine volume. In this slide, you have an extra information about the areas in the nephron and the function of them. Let's narrow the previous information down. Let's talk specifically about the function of the glomerular corpuscle or renal corpuscle that is made by the glomerulus capillaris and the renal corpuscle. The function is filtration only, and that filtration is driven by hydrostatic pressure mainly. Now, these glomerulus capillaris are special capillaries, are called fenestrated. If you remember the three types of capillaries, continuous, fenestrated, or discontinuous, is the difference between each other is the gap in between the simple squamous epithelial cells. In other words, how big is this gap is the one that determines the type of capillaries. In the case of the glomerulus, they are fenestrated. The gap in between those cells is a little bigger than in the continuous capillaries and is a little smaller than in the discontinuous capillaries. But what is important when they are designed in order to increase the filtration from the capillaries into the capsule. And the product in the capsule is called filtrate. The molecules that are inside of the glomerulus need to reach the renal capsule by filtration. And in order to do so, they need to go through different layers, three different layers. The capillary layer, the basement membrane, and the inner layer of the capsule by itself. And pay attention about this word that are the podocytes. The podocytes are special cells that make that inner layer of the capsule and protect and wrap the glomerulus capillaris in order to select the filtration. I mentioned before about the podocyte, and the podocyte has extensions like little hands that are wrapped around, are enclosing the capillary walls. Why is that? Because they ensure that the proteins keep inside of the capillaries. What is important is that fenestrated capillaries, called glomerulus capillaris, the gaps, as I mentioned before, in between the simple squamous epithelium are bigger than the continuous capillaries. But we have to be sure that the blood cells and the proteins don't escape in the filtration. And because of that, we have the podocytes. The podocytes are the ones that avoid the loss of proteins from the bloodstream into the filtrate. If we have that case, it's totally abnormal. Now, in case some of the proteins can escape the bloodstream from the glomerulus into the capsule, the one that has more chances to be filtrated is the albumin, because from all the proteins are the smallest in size. But in normal scenario, in case we filtrate proteins in the capsule, that one are going to be reabsorbed in the proximal convoluted tube. We mentioned that all the molecules that are capable to escape the glomerulus into the capsule, the renal capsule, are called a filtrate. Filtrate or ultrafiltrate is the same, they are synonyms, but the ones that are capable to escape are based on the size. You notice in this drawing that the ones that remain inside of the glomerulus are the little bits that are green color, and these ones are probably the proteins. But remember, we shouldn't have in the filtrate proteins or blood cells. In other words, that molecules and that cells that remains in the glomerulus from the beginning to the end remain with certain amount of plasma. We mentioned before that the forces that driven the molecules to escape from the glomerulus into the renal capsule are the hydrostatic pressure. Now, in previous chapters, we talk about the net filtration pressure, and in this case, in these capillaries are not exemption, is the amount of hydrostatic pressure inside of the glomerulus minus the hydrostatic pressure inside of the capsule. This one is the net filtration pressure, and because of that, the hydrostatic pressure inside the glomerulus is always way higher. One of the reasons why it's way higher is because of the anatomy of the afferent and afferent arteriole. The afferent arteriole is significantly wider than the efferent arteriole and is designed in this way to increase the hydrostatic pressure inside of the glomerulus in order to increase the filtration in from those glomerulus into the capsule. The other one is the colloid osmotic pressure, and the colloid osmotic pressure is the one produced by proteins and are the ones that remain stable. In normal conditions remain stable, and because of that we filtrate water, a significant amount of water, but also that osmotic pressure inside of the glomerulus are the ones that attract or allow to certain amount of water remain inside of the glomerulus. We have an average of 5.5 liters of blood in our system between males and females adults. Now, from that 5.5 liters of blood, we filtrate this amount of blood, an average of every 40 minutes in our day. In other words, we filtrate 180 liters per day under normal circumstances in our urinary system. That one is equivalent to 45 gallons of blood per day. It's the same blood, the 5.5 liters of blood that are filtrated every 40 minutes. Now, imagine that we filtrate 180 liters per day of blood. However, we urinate only an average between 1.5 liter to maximum 2 liters of urine. In other words, that 180 liters per day of blood filtrated in some way, somehow, is reabsorbed and is getting back in our system. The only molecules that we are not going to get back in our system are waste or toxic molecules. How we regulate our filtration? How we regulate our glomerular filtration rate? Now, there is only one way that we can regulate the glomerular filtration rate, and it's only either by vasoconstriction or vasodilation of the afferent arteriole that originate the glomerulus. Only by vasoconstriction or vasodilation. Now, this one is the only way that we have in order to regulate the glomerular filtration rate, but we can adjust the width of that afferent arteriole by different mechanisms. One is called extrinsic, the other one is called intrinsic regulation. The extrinsic regulation or extrinsic mechanism to regulate the glomerular filtration rate is using, extrinsic is outside of that afferent arteriole, outside, away from that afferent arteriole. Extrinsic regulation takes place by the sympathetic nervous system. In other words, if we engage the sympathetic nervous system, we produce vasoconstriction of the afferent arteriole. By doing so, we reduce the blood flow and we decrease the glomerular filtration rate. In intrinsic mechanism, we use a different system, and the system to regulate the glomerular filtration rate by intrinsic regulation is called renal autoregulation of the glomerular filtration rate, by which that afferent arteriole can be vasodilated or vasoconstricted by a special mechanism that takes place right there next to the afferent arteriole. Both mechanisms are necessary to eliminate waste and to regulate the blood pressure, and both complement between each other. It's not one or the other one. For sure, they cannot overlap between each other. It's either the extrinsic or the intrinsic, not both acting simultaneously. I want to be more specific about the extrinsic mechanism to regulate the glomerular filtration rate. This system or this mechanism is used when the blood pressure goes below 80 millimeters of mercury or rise above 180 millimeters of mercury, and always if the intrinsic mechanism cannot balance the blood pressure by itself. Now, very well known that we engage in sympathetic nervous system if we engage in a fight to flight reaction or if we have to engage in exercise. In this scenario, is a system that we helps to divert blood to the heart and muscles and reduce the blood flow in other systems. And how we reduce blood flow in the urinary system is producing vasoconstriction in the afferent arteriole. Producing the vasoconstriction in the afferent arteriole, we reduce the blood flow inside of the glomerulus, and reducing the blood flow, we decrease the glomerular filtration rate. Now, is something that we need to do because in that scenario, we help to increase the blood pressure that is automatically happen when we engage in physical exercise. But this extrinsic mechanism is also used to compensate a fast drop in blood pressure. How is helping? Because produce vasoconstriction. Yes, in case we have that scenario, that um, clinical scenario that the blood pressure reduced drastically, the afferent arteriole is vasoconstricted, and when it is, reduce the urine formation. Reducing the urine formation is the one that allowed us to retain more water. More water in plasma increase the blood pressure. Let's analyze these two scenarios. On the top left, you have a drop in blood pressure. On the top right, you have engaged in exercise. In both scenarios, we need to engage the sympathetic nervous system. Now, the drop in blood pressure is sensed by baroreceptors. And in case of exercise, automatically we engage in sympathetic nervous system, increasing the heart rate and increasing the strength of contraction. Producing the engagement of the sympathetic nervous system triggers three different actions. One is in the heart, the other one is in the kidneys, and the other one is in skin and gastrointestinal tract. What happens in the heart? In the heart, by engagement of the sympathetic nervous system, as I mentioned before, we engage the heart rate and we increase the heart rate and we increase the contractility. By doing so, we increase the cardiac output. Increasing the cardiac output, increase the blood pressure. One problem solved. The other pathway that is used the sympathetic nervous system is the afferent arteriole in the kidneys, producing vasoconstriction. Producing vasoconstriction reduces the glomerular filtration rate. Reducing the glomerular filtration rate reduces the urine production. Reducing the urine production increase the blood volume. We eliminate less urine, less water in urine, and we keep more water in plasma. By doing so, we increase the blood volume. By increasing the blood volume, we increase the blood pressure. And the third pathway is vasoconstriction of the arterioles in skin and gastrointestinal tract. By doing so, we increase the peripheral resistance, and increasing the peripheral resistance increase the blood pressure. So this one is the whole mechanism how we increase the blood pressure if we engage in exercise or if we have a drastic drop in blood pressure. Now it's time to describe in more detail the intrinsic mechanism to regulate the glomerular filtration rate. This one is also called renal autoregulation and is the one that takes place and is used by our physiology most of the time. When the blood pressure goes between 80 to 180 millimeters of mercury, means that is most of the time. And the goal is maintain the glomerular filtration rate at constant level, regardless the fluctuations in the blood pressure, because reducing blood pressure affects the glomerular filtration rate. That is true, but doesn't damage the glomerulus. On the contrary, if the patient is exposed to high blood pressure, that constant high blood pressure in the long run, if it is not neutralized by renal autoregulation, can damage the glomerulus, and is the reason why that intrinsic mechanism tries to keep that glomerular filtration rate in balance to avoid any further damage of the glomerulus. Now, what happens in the afferent arteriole? If the blood pressure drop below the normal, that afferent arteriole is dilated because there are less blood flow inside of the glomerulus, and in order to keep that glomerular filtration rate in balance, we vasodilate the afferent arteriole in order to allow more blood getting into the glomerulus and more filtration, produce more urine, and that glomerular filtration rate remain in the normal rate. On the contrary, if the blood pressure is above the normal, we have to protect that glomerulus from high blood pressure. How we are going to do it? Produce vasoconstriction. Vasoconstriction of the arteriole is the one that reduces the blood flow and is protecting the glomerulus to filtrate too much molecules, and also that molecules that need to remain in the glomerulus, like the blood cells or the proteins. And that one is also called, the outdoor regulation is also called myogenic constriction, because the afferent arterioles are made by the smooth muscle, like any other arteriole or vessels in our body are made by the smooth muscle. That's why it's called myogenic constriction. This intrinsic regulation of the glomerular filtration rate is achieved by one structure that is called juxtaglomerular apparatus. This juxtaglomerular apparatus is composed by two different structures. One is called macula densa, that are special cells inside of the finishing of the ascending limb in the nephrons. In this finishing of the ascending limb at the cortex area, inside we have that macula densa, special cells that are basically, they have stretch receptors. The other component of the juxtaglomerular apparatus are the granular cells, the one that produce renin. Now, in that intrinsic regulation, is intrinsic because, as I mentioned before, it's happening right there around the afferent arteriole. What happened? This afferent arteriole is the one that is bringing blood inside of the glomerulus. Now, how much pressure they bring, it depends. Could be normal, could be above the normal, or could be below the normal. If that blood pressure inside of the afferent, getting into the afferent, is coming with a high blood pressure, then macula densa sense it, because they have stretch receptors, sense it that the afferent arteriole is too much distended. And in order to protect the glomerulus capillaris, produce a vasoconstriction. How is vasoconstriction of the afferent arteriole? Why? Because we need to reduce the blood pressure inside of the glomerulus to keep that blood flow in normal rate. Now, how the macula densa communicate with the afferent arteriole is through chemical molecules that inform the afferent arteriole that they need to produce that vasoconstriction. On the contrary, if that afferent arteriole is bringing blood with low blood pressure, that afferent arteriole is vasodilate. But keep in mind that that intrinsic regulation takes place between the juxtaglomerular apparatus and the afferent arteriole. The communication between the juxtaglomerular apparatus, specifically the macula densa, with the afferent arteriole is called tubuloglomerular feedback, and is because there is a communication between that macula densa with the afferent arteriole. This slide is explaining pretty much the same I explained in the previous slide, when there are different scenarios that stress receptors in the macula densa are sensed if the blood pressure is normal, if the blood pressure is above the normal, or if the blood pressure is below the normal. And according with that information is communicating with the afferent arteriole what is the step to follow. Is vasoconstriction to reduce the glomerular, to reduce the is vasoconstriction of the afferent arteriole to reduce the blood flow inside of the glomerulus, or is low blood pressure and we need to vasodilate to increase the blood flow in that particular glomerulus. But all these mechanisms happen in order to keep the glomerular filtration rate in balance regardless the fluctuations in the blood pressure. So, the two mechanisms that help to regulate the glomerular filtration rate are the intrinsic, produced by autoregulation between the juxtaglomerular apparatus and the afferent arteriole, or the extrinsic mechanism that take place using the sympathetic nervous system.